Hi students, I would like to continue the lesson chromosomal basis of inheritance. And this one, today, we have the topic how to calculate the recombination frequency. First of all, what do you mean by recombination frequency, or simply what is called RF, recombination frequency. So it is a percentage of recombinant progeny in a crossover. Is called recombination frequency. The percentage of recombinant progeny in a crossing over or a crossover is called what is known as actually recombination frequency. Now it can be calculated by using the following formula. Now I am taking the data obtained from the alleles of coupling reaction or coupling configuration. Now, if I am taking the coupling configuration with reference to the sweet pea about the color of the pollen grain, the shape of the color, the shape of the pollen grain. If I am taking the color of the pollen grain, we have purple. And another character, the shape long. We already see out of 100 we have received 44 progenies. Then we have purple, what is called round. We have 6. And then we have what is called red, long. We have we have 6. Then red, round, 44, totally 100. So this is the thing what is in already. Then how to calculate what we have, the recombinant frequency or percentage recombination frequency. Now, recombination frequency can be calculated by using the following formula. So, this is the number of recombinants divided by, that is total number of offsprings. You can write either total number or simply number of offsprings. This is the formula normally used to calculate what is called recombination frequency. Now out of these 100, if you are taking what we have the coupling configuration, so the data I would like to take to discuss about recombination frequency, nothing but the coupling configuration. What is studied in the case of sweet pea, Lathyrus odoratus, just proposed by Botany and also Punnett. So these are the two parallel combinations. We started with a cross, that is purple long and then red long. The remaining two are the recombinants that were formed just because of the crossing over. So the number of recombinants totally, that is what we have 12. So if we are taking this one, number of recombinants 12. So these are the new varieties. These two are the parallel combinations and these two are what we have recombinants totally. So out of 100, the recombinants are 12. Then for 100, we are finding out the percentage of recombination, that's called recombination frequency. Now the percentage of recombination or recombination frequency is equal to 12%. This is a way you can calculate for any value that is given. So, number of recombinants divided by number of offsprings multiplied by 100. So, by this way you can just find out the percentage of recombination or recombination frequency. We have different units, we'll see that one. So, percentage recombination in terms of what is called 12%. If you want to represent the same one in terms of frequency, so out of 100, 12, so 0.12. This is called also recombination frequency. So either we can represent what's called the percentage of recombination, or we can represent in the form of recombination frequency, that is out of 100, 12, so we have the value is 0.12. This is one. Okay, now we have to go. A diagrammatic representation is given in the book. And based on the diagram, you have to calculate the recombination frequency. Here is given two types of figures. Figure A representing there are about 49 meiosis or meiotic divisions with no crossing over. The second one B, it represents one meiotic division with a single crossing over. You know that one, as a result of one meiosis, we have received 4 cells or 4 progenies. 4 cells or 4 progenies. And as a result of one meiosis, we have received 4 cells or 4 progenies. And here I represented what we have 49, the number. The numbers representing what we have, the number of what is called the progenies formed in each meiotic division. The number of progenies formed in each meiotic division. For example, one meiotic division that is normally produces 4, but here there are 49 meiotic divisions. So the total number of what is called the progenies or the offsprings produced because of 49 meiosis is equal to 196. That is 49 into 4. Here I represent only 4. You have to multiply these 4 with 49. That's why I represent 49, 49. So one meiotic division produces 4 offsprings. But here it is given 49 meiotic divisions. So the total number of progenies, or we have the offsprings produced because of 49 meiotic divisions, 49 into 4. Okay, so I am taking here now. So number of progenies produced in one meiosis is equal to 4. You know that 4 sperms are produced during gametogenesis. Like that, here we have to consider 49 meiotic divisions. In each division we have produced 4 progenies. That means you have M1s. Now the next one, number of progenies produced in 49, that is what is called meiotic divisions, 49 meiosis. So this is equal to 49 into 4. So we are getting what is called 196 offsprings. 196 offsprings. This is number 1. Here there is no crossing over. No new recombinants are produced. No new recombinants are produced. All are actually similar to the parallel types. Now let's take this one. Here there is one meiotic division. So one meiosis. The second one, this is what's called A, and this is taking adjacent B. One meiosis produces, once again, is actually 4 progenies. You'll take that one in the form of offsprings. As I'm using the word offsprings, I'm using the same word also, 4 offsprings. Okay. Now the total number of offsprings produced, total number of offsprings, or we can say the progenies, total number of offsprings produced. I'm using either progenies or we can say what is called offsprings, both are same. So here 196 plus 4, that is 200. That is total number of offsprings produced in 50 meiosis. Because the previous one we have 49, this one just actually what we have, the 50th one. So total number produced is equal to 200 progenies or offsprings. Now out of these 200 progenies, you can see only 2 of them are recombinants. 1 plus 1. I represent in green, 1 plus 1. So only just 2 of them are recombinants. So we have to take the recombination frequency. I am taking this one. So RF is equal to number of recombinants divided by number of offsprings. Number of offsprings. Then multiplied by 100. Multiplied by 100. Now we have to just substitute these values. Now the total number of offsprings is equal to 200. The total number of offsprings is equal to 200. Out of 200, only 2 of them are recombinants. You see the number of recombinants is equal to only 2. You see that 1 plus 1, so together 2. Then for 100 is equal to, you know that 1%. So the recombination frequency for these illustrations is equal to 1%. I repeat once again, there are 49 meiotic divisions. In one meiosis, 4 progenies are produced. So 49 into 4, 196. Here another one, the 50th meiosis. Here also 4 progenies are produced. So the total number of offsprings or progenies produced is equal to 200. Out of 200, you can see only 2 of them are what we call the recombinant varieties. So out of 200, only 2 are recombinants, and for 100, now the recombination frequency is equal to 1%. If you want to represent the same percentage for 100, it can also be represented as 0.001. This is for 100. 0.001. This is for 100. So this is about what we have the calculation for the diagram what is given in the book. Okay, I represent simply 49, 48 just to understand the fact only. Not each one is 49. In each meiosis, we have just actually 4 individuals. There are 49 meiotic divisions. Likewise, you have to calculate that one. Okay, thank you. We have to go for some other problems later. Now the next one, what is genetic mapping? In a chromosome, the genes are arranged in a linear order along the entire length, just like this, A, B, C, D, like that. This is called the linear arrangement of the genes. And each gene has a specific what's called location. The specific location of the gene is called actually locus. Suppose this is what's called the A locus. Each gene has a specific position on a chromosome. That region is called what is known as a locus. Now what is genetic mapping? So it is nothing but the diagrammatic representation of the position of genes. Number 1, the position of the genes. Diagrammatic representation of position of genes. You know that one, A in the first position, B in the second position, C in the third position, like that. So it refers to what we have actually the diagrammatic representation of the position of the genes. First, second, third, like that. And the related distance between the adjacent genes. The related distance. Now this is what is called the distance, related distance between the genes. And this is called genetic mapping. A diagrammatic representation of what we have the position of the genes in a chromosome and the related distance between the adjacent genes. How far they are placed, either closely or far away. And that is called what is known as a genetic mapping. Now, how can you just calculate this one? Normally, the genetic mapping is also called the linkage map because the genes are arranged in a linear way, linked together. It's also called linkage map. Now, this genetic mapping is directly proportional to, or we can say genetic map is directly proportional to the percentage of recombination between the genes. The percentage of recombination between the genes. So, what we are using the unit, the recombination frequency. A percentage of recombination or crossover value, and based on this only we can just construct the genetic map. So the unit or what we need a factor that is nothing but the recombination percentage of recombination frequency or crossover value. These are all required to construct what is called a genetic map. Now it was first developed by one of the students of T.H. Morgan by name Alfred H. Sturtevant. He was the first person developed this concept, what's called genetic mapping. He was a student of T.H. Morgan. So, this genetic mapping normally provides a clue about the genes, where the genes lie on the chromosome. It gives a clue, it provides a clue about the location of the genes on the chromosome. So, this is about genetic mapping. Then what is the unit of distance? What's called map unit distance. Map unit distance. So between A and B there is a distance. How can you measure this one? What is a unit? It is normally used just for what is called the mapping distance. I'll take that one now. In normal conditions, we are using, you know that one, the different units, the SI units, centimeter, millimeter or meter, to just measure the length of a particular object. But in the case of what we have map distance, it refers to what we have, what unit we are using in a genetic map. So, the unit of map distance in the genetic map is called map unit. Instead of using centimeter or millimeter, we are using what's called map unit, simply MU. Okay, so in a genetic map, the unit of map distance is called map unit between the genes, for example between A and B genes, like that. I represented here for example 3.5, that is map unit. This is a distance between A and B, 3.5, what is called just actually map unit. Instead of using centimeter, millimeter, etc., we are using what's called a map unit. So the unit of map distance is normally called a map unit. So one map unit is equal to 1% of crossing over. So the map unit is based on the percentage of crossing over. If you have one map unit, then 1% of crossing over. If you have two map units, it is equal to 2% of crossing over. If you have, for example, 10 map units, we have 10% of crossing over. Now, one map unit is also called 1 centimorgan, in honoring T.H. Morgan, because he was the one just actually gave a meaning for what is called just genetic mapping. He is one of the students who only developed what is called the mapping, genetic mapping. So one map unit is also called 1 centimorgan, in honoring T.H. Morgan. 100 centimorgan is equal to 1 morgan. This is what we get the distance calculation. So it may be in the form of 1% crossing over, or 1% recombination frequency, or we can say like that, 1% recombination or recombination frequency. We have different methods. I will come to this right now. So, for example, the distance between A and B genes is equal to 3.5 map units. So 3 map unit is equal to or equivalent to 3.5 centimorgan or 3.5 percentage, recombination percentage, or percentage of recombination, or in terms of frequency 0.035, that is recombination frequency. This is because we have 3.5 for 100, then we are getting what is called 0.035. So we can use different measures, maybe in the form of map unit, or in the form of centimorgan, or in the form of what is called percentage of crossing over, or in the form of recombination frequency. So anyway, one map unit is equal to 1 centimorgan. 1 centimorgan is equal to 1% crossing over, or it is equal to just what we have, recombination frequency. So here it is given 0.05, because for 100 only we are calculating. For 100 if it is 1, so 0.001. Okay, that's about. So the distance between A and B genes here in the problem given 3.5, that is map unit, is equal to 3.5 centimorgan, because one map unit is equal to 1 centimorgan, or it is equal to 3.5 crossing over value or crossover. That's the percentage of crossing over. Or we have the recombination frequency in terms of what is called the fractions, 0.035. So between the genes, now I am taking for example 4 genes, A, B, C, D. This is what is called the genetic map. So what is the distance between A and B as given here? 3.5 map units or centimorgan, or 3.5% or 0.035. Now the distance between B and C is equal to 7.5 map unit, and similarly the distance between C and D, 5.5 map unit. So from this one you see that one normally in a genetic map one can find out the order of arrangement of genes. Okay, the order of arrangement of genes as well as the distance between the genes. So between A and B we have 3.5. So anyway, this is a diagrammatic representation actually showing the distance between the genes as well as the order of arrangement of genes. Here the order of arrangement of genes is equal to A, B, C, D. The distance between A and B is equal to 3.5. The distance between B and C is equal to 7.5. Now how can we construct a genetic map? A genetic map or a map distance, normally genetic map with the distance as well as also the arrangement. A genetic map can be constructed just based on the results of what we have, the test crosses with a pair of genes or for pair of genes. If you are taking for example just 2 alleles, this is what is called heterozygote, we are taking only 2 alleles. So this is what's called a dihybrid, or we can say double recessive heterozygote. Double recessive heterozygote. So double recessive heterozygote is crossed or test crossed with a double recessive homozygote. And this is called what is known as two-pointed test cross. If we are taking only 2 alleles, then it's called two-pointed test cross. If we are taking 3 alleles, then it's called three-point test cross. Okay, so genetic map can be constructed from a series of test crosses for pairs of genes, and this is called two-point test cross. Simply two-point cross in which we are crossing what we have a double recessive heterozygote with a double recessive homozygote. Double recessive heterozygote and double recessive homozygote. Now, it is not effective. What we have, the two-point test cross is not effective. It is not efficient because in this one we cannot detect what is called the double crossing over. We cannot find out the double crossing over. That's why I mentioned here. This is not efficient because the double crossover is missed. So we cannot detect the double crossover to enable to find out the accurate map distances. Another cross is so efficient that is called the three-point test crosses, where we are taking 3 alleles. 3 alleles. So two-point test cross involves only 2 alleles. The next one, what we have, three-point test cross involves 3 alleles. If you are comparing what is called two-point test cross and then three-point test cross, the most efficient one in constructing the genetic map is three-point test cross, because in that one you can find out the double crossover. So double crossover can be detected, which will give more just accurate what is called the map distances when compared to this one. Here we cannot find out the double crossing or value, that's why it is being neglected. The most accurate one is three-point test cross. We have to go the next one. Okay, this is about what we have, the unit of map distance. Okay, unit of map distance is called a map unit. One map unit is called 1% of crossing over. It is also equal to 1 centimorgan. 100 centimorgan is equal to 1 morgan. So if you are taking just a 3.5 map unit, then 3.5 centimorgan is equivalent to this one, or 3.5% of crossing over, or we can have 0.035 recombination frequency. All are equivalent. Okay, I'll continue about what we have the three-point test cross.